All right, I think we're ready to get started. Good morning, everyone. My name is Jason Peralt. I will be your moderator for today's morning session. For those of you who I have not met, I'm a fourth generation hop grower from the Yakima Valley and a hop breeder with Yakima Chief Ranches and Hop Breeding Company. I am pleased to be kicking off the presentations of the week. This morning's sessions will focus on two areas that are very important to all of us in the hop and brewing industries: hop breeding and federal issues. A quick note for everyone, during the sessions, if you have questions, please add them to the Q&A at the bottom of your screen. And before we get started, I'd also like to thank our platform sponsor for the morning, which is Hop Steiner. With that said, our first topic of the day is one that is near and dear to my own heart, which is hop breeding. With us, we have the breeders that make up our USDA hop breeding team, Dr. Kayla Altendorf and Dr. John Henning. They will be giving us an overview of the history, processes, and strategy and insight into future trends in USDA hop breeding, followed by an open discussion and Q&A session. Here's a little background on the presenters. Dr. Kayla Altendorf actually came to the Yakima Valley via a similar pathway that many of our French Canadian families came to the Yakima Valley. That she came from Minnesota. She earned her MS and PhD degrees in applied plant sciences with a focus on plant breeding and molecular genetics from the University of Minnesota. And she started as the USDA-ARS research geneticist in May of 2020. She's currently based at the IREC in Prosser and is initiating a new USDA breeding program, collaborating closely with the program in Oregon. Her goal right now is to seek to develop improved publicly available hop varieties that benefit stakeholders at all steps of the supply chain. And she's looking forward to stakeholder input in this session, providing invites you to view pre-recorded HRC talks to learn more. Dr. John Henning really needs no introduction to those of us that have been in the industry for a while. John has led the USDA-ARS public hop breeding program since taking over from Dr. Al Hunold in 1996. And he is the lead scientist over USDA's hop research. Dr. Henning is an internationally recognized hop geneticist working on the forefront of hop research, covering genomics and molecular breeding. Currently holds a courtesy professor appointment in crop and soil sciences, along with an affiliate appointment at Oregon State University's Center for Genomic Research and Bioinformatics, and collaborates closely with genomics and bioinformatics faculty at OSU and elsewhere. He originally comes to us from studying at UC Davis and came to hops after a short stint at New Mexico State University, focusing on alfalfa research. So with that, look forward to getting going on our talks here, and invite, and as we move forward, I'd like to ask the audience to consider some questions as they present their information. Some of the more common questions that would be covered would be: what would you like to see the public hop breeding programs do? What role can the public hop breeding programs play within the industry? What can they do to improve? And what problems have you observed in the last few years that you think they should work to address? So just keep those in mind as we move through these presentations. And with that, I invite Dr. Kayla Altendorf to get us started.
Actually, I'm going to start first, Jason.
Right,
we switched it up on you. Anyways, boy, I'd sure rather be doing this meeting in person and be able to see each one of you, but unfortunately, the way things are, it's the way it is. So I'm coming to you from beautiful downtown Albany, Oregon. And today what I'll be covering is basically the history of hops from the standpoint of its evolution and how we came about to have hops, and then looking at some botany as well. Then we're going to be looking at what are the selections and the traits that are important for plant breeding, and particularly what we in the USDA are trying to emphasize in our breeding program. Then it's going to be followed up by Kayla, where she's going to be talking about the history of the USDA hop breeding program, as well as what's the pipeline for developing a hop variety. So looking at hop botany, the species and the scientific name is Humulus lupulus L. and it's actually the variety Lupulus. Hops arose, or they're part of the family of Cannabaceae, which many of you realize is partly, it shares with cannabis. The line that we actually grow for hop growing is Humulus lupulus L variety lupulus. There are other species and subspecies or varieties, one of which would be the wild Japanese hop, which is Humulus lupulus cordifolius, or the wild North American hops, which are Lupulus variety lupuloides, neomexicanus, and pubescens. But there's some other species too, the Humulus japonicus and Humulus unionensis. Hop is a diploid crop. What that means is it has two sets of chromosomes. The total number of chromosomes present in hop is 10, but since that's doubled in each cell, there's 20 chromosomes. And you can see the little graphic down at the bottom showing the different hop chromosomes. Of these 10 chromosomes, 9 of them are what we call autosomes. And what that means is they're chromosomes that are not involved in determining the sex of the plant. In addition, there's the X chromosome and the Y chromosome. XX would be a female, XY would be a male, just like we have in humans and animals. So as I mentioned, there's male and female plants, and this is called dioecious. And this is somewhat of a unique plant specification in that not all plants have both male and female plants counterparts. Most of them, in general, have all of their flowering pods on the same plant. The female inflorescence, or the hop cone itself, is actually what's harvested for production. Cones are used primarily for beer bittering and flavoring. And what makes the hop cone so special are the things called lupulin glands. The lupulin glands are the power pack points where all of the flavoring compounds are developed and stored. The other aspect of flowering is that it's strongly controlled by photoperiod, meaning that the length of the day is whether or not a plant is going to be flowering or not. So the farther north you go, you have higher production because you have a longer day length. And on contrary, in the southern part of our hemisphere, the longer the day, the greater the amount of flowering that's going to be taking place there. So each hop variety is a clone from the original mother plant. And in this picture right above, under crowns and rhizomes, I have a picture of a mother plant. And what you see there are a whole bunch of what look like roots, but what we call them are rhizomes. Rhizomes actually contain these adventitious shoots that will become the plants. And so when you cut one of these rhizomes off and put it in the soil, a new plant is going to be derived from it, but it will be an exact clone of the mother plant. Hops they're not planted by seed, except for in breeding nurseries, and there are no hybrid varieties. Hops are actually produced in general on 6-meter trellis. There is some cases where it's produced on a 3-meter trellis, but that's a different production system. For the most part, what we do here in the USA is grow on 6-meter trellises. Looking at some of the flowering of hops, we have two forms of flowers, one being the perfect flower and one being an imperfect flower. Perfect flowers contain both the male and female parts present on the same flower or within the same flower. Hops fall into the imperfect flower section in that they either possess a male flower or a female flower that are distinct from each other. And 5% of all plants are what's called monoecious. And if you look over to the right, the one we're most familiar with is corn, where their tassel is the male flower and the ear is the female flower, but they're on separate parts of the plant. Hops fall into what we call the dioecious, as I've already mentioned. And this is a situation where Each plant is either a male or a female, so it should only have male flowers or female flowers. Looking at hop evolution then, the theory is that hops, or the variety, excuse me, the humulus actually arose from evolution from cannabis. And the reason we think this is the case is that the genome of cannabis is around 800 megabases, whereas humulus, it's almost 3 times that size. And so we believe that there was a genome replication that occurred from cannabis to humulus. From humulus then, we believe that Humulus japonicus was the first to be evolved, and then from that, Humulus lupulus arose from that particular species about 6.38 million years ago. And there are 3 evolutionary stages, or if we will, sort of routes for Humulus lupuloides that occurred about 0.5 to 0.7 million years ago. The Asia to North America route produced the varieties Lupuloides, Neomexicanus, and pubescens. In Japan and southern China, Cordifolius arose out of that. And then, of course, the most important of all of these, transfer out to Europe, was the development of the variety Lupulus, which we grow for hop production. And this is just sort of a graphic showing the movement of hops through the years. And the center of origin is pretty much assured to be central China. And if you look, the yellow line transfers from central China to North America. And from there, we get our Neomexicanus, pubescens, and Lupuloides. But there also was a transfer from central China to Japan, which rose the Humulus japonicus. And then the pink line went from China over to Europe, where the Humulus lupulus variety Lupulus evolved and arose. And then from there, not necessarily an evolution, but certainly a transport. Central Europe, hops went to Great Britain, and then from Great Britain and from the Central Europe, they were transferred over to New York first, and then from New York, they went to the Midwest and Wisconsin, and then from there to California, then up to Oregon, and then finally up to Washington and Idaho. Much of all of these transfers in the United States were dependent upon presence of disease or the realization that higher yields could be obtained in different locations. So what are the breeding goals of hop breeders? Obviously, one of the first and foremost is that of yield. And yield, when you're selecting for yield, there's actually multiple factors that go into play for that. What's the overall plant vigor? Is it a real vigorous hop plant? Lots of biomass produced. What's the cone distribution all along the hop plant or the axis of the hop? Are the arms grape clusters? In other words, just big masses of hop cones, but not necessarily long arms. And then again, what's the shape of the cone and the density of the cone? And on the right there, you see a picture. This is actually a single string for a hop plant, one of the breeding nurseries, that shows great distribution of hop cones all along the axis of the plant, with long side arms and lots of flowers on each one of those side arms. So this is sort of the ecotype that we're shooting for. So when we also begin breeding and making the cross and deciding what sort of a cross we're going to make, the goals are we going to develop a super alpha, or are we going to try and develop an aroma line? And if it's an aroma line, is it sort of a citrusy type? Is it tropical fruit, resinous or piney? If we're looking at super alphas, sort of the major goal there is to develop something that has 20% alpha and 20 bales per acre. Then, of course, we need to figure out what's the usage of that particular hop. Is it going to be used for lagers or pilsners, or is it going to be more of an IPA style hop? Get back here. Yep. Okay, keep doing that. So flavor is obviously one of the biggest components that we have to be looking at when we're breeding for hops, new hop varieties. Great brewing characteristics are required. The competition is so strong out there for new hop varieties that you have to have something that's new and unique that's going to grab the attention of brewers. I've had several hop experimental lines that have easily had over 3,500 pounds per acre, but the flavor when they were brewed with just was not good. And the result is that that hop never leaves the experimental yard. I do keep those for breeding purposes as potential yield increasers. But when we're talking about flavoring, there's several components that we have to look at. The one that makes hops used in beer is that of bittering acids. The alpha acids and the beta acids. These are the components that actually make beer bitter and give it its punch that you expect when you're drinking a beer. Essential oils come into play as well. There's too many of these to talk specifically about. The major ones that are almost always found in each one of the hop lines are myrcene, humulene, and caryophyllene as essential oils. But the important thing to note is it's not necessarily a single compound that makes a hop a great hop. It's actually the combination of all of the essential oils that make up that hop that give it its distinctive flavor. There are some people that believe strongly that sugar-bound essential oils are actually more important in that the essential oils themselves actually will boil off during the brewing process. And so only those sugar-bound oils are what's left over in giving the flavor. Now, last but not least, we have to talk about thiols, because thiols are potent sulfur-bound compounds that just a small amount will actually produce great flavoring. And some of these things, as we've heard of, like 4-MMP and other compounds, do produce flavors that are very unique and extremely desirable for hop production, or excuse me, for hop brewing. Breeding also has to take a look at the plant morphology. And as I mentioned, how are the cones distributed along the axis? Are the arms really long? Are they 5 feet, 3 feet, 1 foot? Are there a lot of flowers that are along that whole arm? So these things all come into play for whether or not we're going to actually choose a hop variety. And top heavy, bottom heavy. If a hop is too top heavy, sometimes that can tend to plug up a picker, and so we don't want that. If they're bottom heavy, they can get lost in the process of cutting a hop plant. So we want the hop cones to be present all along the axis. Height is another aspect. There are hop varieties around the world that are considered dwarf varieties, and in Great Britain, their hop production is dependent upon dwarf hops. These are ones that actually have shortened internodes and a determinate growth stage, and they're grown on a hedge rather than a trellis. So we can't talk about hop breeding without talking about breeding for plant disease resistance. Two of the diseases that I'll talk about today are the most prevalent and most that hop growers have to deal with. The first one is powdery mildew that's caused by Podosphaera macularis. And this particular obligate parasite is found across the United States, but is predominant in hot, dry climates such as the Yakima Valley or the southern Idaho. It's controlled via spraying and the growing of resistant lines. There are 7 known sources of gene-for-gene resistance, which does provide a complete resistance to powdery mildew. However, this particular style of resistance does break down relatively quickly. And many of you are familiar with the variety Nugget and a number of other lines that were derived from Nugget has what they call R6 gene. That has just within the last several years, the resistance has broken down to that. And so now we're looking for different resistance sources. So as the public breeding program and the private as well, we're looking at combining R genes from multiple sources in the hopes that having multiple R genes present in the same background, this will result in providing a stronger resistance. We're also looking at quantitative resistance. And now this is just a resistance that isn't complete, but it does give the plant a strong uphold, and the colonies don't actually take off on the hop plant itself. Downy mildew is another disease that the USDA is extremely concerned about, and part of the reason for that is that the varieties that we produce and try to focus on are meant to be grown across the United States. And so powdery mildew is primarily found in warm, humid areas such as Oregon and the Midwest, the Northeast and the Southeast. And so this is a real problem in these areas. At this point, it's controlled via spraying primarily and the growth of tolerant lines. There are no known R genes or gene-for-gene resistance. We have partial resistance, but most lines are susceptible. And so this level of resistance is simply a determinant of how often you're going to have to spray. If you have a tolerant line, you don't have to spray as much, but if you have a susceptible line, you're going to be spraying quite a bit. Breeding efforts are focused now primarily on population development, and these are just good old-fashioned grunt, select the best offspring, cross them over again, and then do that selection again and just keep crossing until you have a population that has good resistance or quantitative resistance to downy mildew. So the other area that we need to be focusing on as breeders, and I think this is something that the future is going to be directing more of, is that of breeding for resistance to hop aphids. The hop aphid is Pherundon humuli, and it's primarily a problem in spring and early summer. However, you can get late season infestations, and if you do, that can actually ruin your crop because the aphids leave honeydew over it and mold grows in that honeydew, and the cones themselves get moldy and the whole lot gets thrown out. Hop aphids prefer cool climates. Now, one of the things that I think a lot of people don't know is that the alternate host for hop aphid are your peaches, pears, and plums, and members of that same family. And so these aphids overwinter on those crops. So if you've got those particular crops right next to your hop yard, you're actually introducing a big load of hop aphid into that yard. They're fairly well controlled with sprays, and there is one report of a gene for resistance in Great Britain, although we don't have that necessarily gene located here. Another problem pest that we have to deal with is two-spotted spider mites, and these are nasty things because they can pretty much grow and attack anything that has green on it. They have multiple alternative hosts. They do prefer hot, dry conditions for infestation, and they can defoliate a plant and destroy a crop. And you see in the far right a picture of a plant that got hit real hard with spider mites, and you see the cones are all browned and ready to dry off. There is some resistance reported in some of the USDA hot mail lines that we do have, but I think the biggest point is you need to have timely prophylactic spraying right before harvest or even earlier than that to prevent infestations of this pest. So we also have to take into account what's the farmer's perspective. How well does it train? How well does it grow on the string? Hop vines actually have hop hairs or trichomes that actually help it grow up the string. And in some of our crosses that we make, you may end up with a hop vine that doesn't have any of these hop hairs, and so it'll start growing up, but then something happens, a wind a little bit, and it just falls right off the string. And so typically what I try to do is if that's a problem, then I'm not going to release something like that simply because it's just going to fall off and never get to harvest. But the other aspect that we have to take into consideration is what is its pickability? How well does it pick? Does do the cones come out nice and clean? Is there a lot of chaff that comes through in there? Does the vine itself have a lot of hop cones that stick to it because it's just taking too much force to remove the hop cones from the vine? So a lot of these things are noted when we're making our harvest and can come into the decision-making process. Finally, another aspect that we breeders constantly worry about is how long does the alpha acids remain in storage? And so this is called storageability. Some cultivars are actually better at retaining their alpha acid than others. And this is sort of tested by a 6-month test at room temperature. So it's a long drawn out test. There's a hop storage index, which is sort of a numerical number that we can sometimes use to get an idea whether or not it's going to store well. But I think all of us are in agreement that molecular markers would be great to be able to select for this trait, just simply because it's so difficult of a trait to predict. So this is the point of the talk where I do some shameless promotion of my 2 latest varieties. And so the first one I want to promote is that of the 2010 008. This is a cross that I made in year 2000 onto Nugget. Currently, it's being grown at Crosby Farms and Poudrebaugh Farms in Washington and Oregon. It's a very early high-yielding aroma line. Oregon, it was harvested this year at August 20th, and Washington, August 23rd. There's no difference in the aroma between those two. And I've actually harvested it earlier than August 20th. But the biggest point about this is it really aids farmers by extending the harvest window. This particular hop line is very floral, very fruity, noble hop style. And there's approximately 3,000 pounds of pellets available. The Brewer Supply Group, Hollinsberry, Mill 95, and Crosby Hop have this currently as pellets. The next line is the one that I made a cross I made in 2006 on Pearl, actually a tetraploid Pearl. So it's a triploid line known as the 074. This is being grown at Siegel Ranch and also at Capitol Farms.
No.
There are it's a very high yielding, vigorous, seedless aroma line.
No.
And sorry, I have a 6-month-old puppy. The brewing reports are all extremely favorable for this. It has really broad adaptability from Washington all the way to Vermont. It also appears to have drought and heat tolerance, and there's approximately 3,000 pounds of pelleted lines available. And these are also found at Brewer Supply Group, Hollinsberry, Mill 95, as well as Crosby Hops. So I want to thank everyone for listening to me. And at this point, I'm going to hand over to Kayla. Kayla will be discussing the history of the USDA hop breeding program, as well as what are the steps that are taken to develop a new hop variety. Thanks.
Change your presenter view, Kayla. Thank you.
Okay, good morning, everyone. Thanks so much for the introductions. As John mentioned, I'll be going over the hop breeding methods, a little bit of history about the USDA hop breeding program, and kind of outlining the pipeline that we utilize for developing new hop varieties. So first method that we utilize to develop new hop varieties is mass selection. So this was one of the first methods used to develop new hops. And in the figure, I'm showing a field of a certain hop variety or maybe a selection. And breeders or kind of farmers early on would identify a plant that did exceptionally well among those plants. And this plant might be a little bit different based on a mutation that arose spontaneously, or maybe was an open pollinated seedling from that line. So breeders would identify one that did really well and then propagate that out as a new variety. And an example of a hop that was produced this way is Yakima cluster. The next method to develop a new hop is utilizing open pollinated breeding. So in this case, you select a female that you think does really well, and you allow it to open pollinate. So in this case, you have limited control, or you have no control over what male pollen actually fertilizes the female plant. So really you're applying selection only on the female in that case. So from an open pollinated cross, you'd harvest the seeds, plant them all out into little seedlings, and you'd select, we'll just say in this case, one individual from that family that did really well. And from there, you'd propagate it out onto larger scale trials and then eventually release it as a new variety. And Cascade was produced using open pollinated breeding. The third approach is pedigree based. You identify a female plant that does really well, and you also select a specific male that you think would combine really well with that female. And again, so in this case, you have intentional selection going on in both the male and female parents. And the same process here, plant out the seedling, select one that does really well, increase acreage and testing, and then eventually release as a new variety. And Centennial is an example of a hop that was produced using pedigree-based selection. And then one final approach is using polyploid breeding. So a common approach in plant breeding is to double the chromosome number. So John talked about this a little bit in his talk, but this has been a fairly popular method of breeding hops. So you would double the chromosome using a chemical mutagen. And I'm showing the hop cone increasing in size because plant parts tend to increase when you double their chromosome number. And you would cross this by a diploid male and evaluate triploid seedlings. So in this case, you plant them all out, select one that does really well, and increase scale and testing, and eventually release as a new variety. And Willamette was a hop that was produced using polyploid breeding. And some of these approaches are used in combination with one another. So you might develop a polyploid parent and then allow it to open pollinate, or you might develop a polyploid parent and intentionally cross it with different males as you would in pedigree-based selection. So a little trivia, if we were in person, this would be a little bit more exciting, but feel free to submit to the chat or just think to yourself. So which breeding method is the most common for USDA released hop varieties? Is it mass selection, open pollinated, pedigree-based selection, or polyploid breeding? And just as a side note, and when I refer to USDA released hop varieties, I'm specifically referring to female hops that were released for commercial use, not for breeding or ornamental purposes. So moving on to the answer, the most popular method of hop breeding is in the USDA hop breeding program has been pedigree-based breeding. Following that is a combination of polyploid and pedigree breeding, like I mentioned, where you would have a tetraploid female crossed with a diploid male, and then a diploid known male, and then select amongst that progeny. That's the second most popular method, followed by open pollinated and polyploid and open pollinated combined together. I'm going to talk a little bit about the history of the USDA hop breeding program. So here I have a timeline. Obviously, this is just a really brief history and not comprehensive in any way, but just to give you an idea of what the focuses have been over the history of the program. And on the top, I have the different breeders that have led the program over the years. So the program was initiated in 1930. There were a few breeding efforts or hop research efforts that preceded this, but didn't amount to much in terms of, at least in the documentation. So the USDA hop breeding program began in Oregon in 1930, and the major focus at the time was on finding resistance to downy mildew. Moving on to the 50s, there was started to be a little bit more intentional selection on the parents, so utilizing that pedigree based method, and also testing individual females with different males to try to get an idea of the breeding value of certain males. And that's a process known as progeny testing. In the 1970s, there was a major focus on combining high alpha acid and high storage stability lines, as well as the polyploid or triploid breeding strategy became popular in that time. In the 80s, there was an increased focus on aroma varieties, resistance to verticillium wilt and downy mildew, as well as early maturity. And then when John took the program over in the 90s, I believe late 90s, he started implementing more quantitative and molecular genetics, and then demonstrated that it's really important to focus on crosses using parents that are distantly related. And then in 2020, we're focusing a lot on QTL mapping. John's been working for a while on sequencing different hop genomes. And then one major breeding objectives that John outlined is disease resistance. And then also there's been some work, and I suspect there will be more work focusing on abiotic stress tolerance. So things like drought stress and heat stress. So on average, from the time of a cross in the USDA program to the time of release, so the number of years to release, takes on average 13.7 years. And you can see here on the y-axis, I have the frequency, and on the x-axis, I have number of years, and the vertical green line indicates the mean of the distribution. So you can see in some cases, it takes upwards of 25 years from the time that the cross is made to the time that the variety is released, but in some cases, it's under 10 years. And I think the method of the breeding that is utilized, as well as the type of variety, all plays a role in this. And also consumer demand and interest too. In some cases, breeders will sit on material until it generates interest. I want to outline the hop breeding pipeline that's used in the USDA breeding program. So in year one, we select our parents and make crosses. And we try to do enough crosses to end up with about 25,000 seedlings. So to make a cross, we select our parents. This is a picture of me actually choosing parents this past summer for my first round of crosses. And John explained this process. You know, you might select your parents based on whether you want aroma family or a bittering family, or if you want to incorporate disease resistance. And then looking back, this binder is something I inherited with the breeding program here in Prosser, which outlines the pedigrees of all the old germplasm in the breeding program. So anyway, you want to confirm that they're not super closely related to avoid inbreeding depression. So there's lots of things to consider when choosing parents, but those are just a few. So in June and July, you collect male pollen. And this is a picture of a male plant in the male yard at IREC. You can see the Horse Heaven Hills in the background. So you cut off some flowers, let them dry out a little, and sieve out the male pollen. And you can actually store this at least for up to a year, which allows you to make crosses with females that mature earlier than the males in the next year. Because in my case, this first year, I was limited by pollen availability based on when the female was ready to be pollinated. So you have your parents that you want to cross. You have to monitor that female plant throughout the summer to determine the optimal time to make a cross. So you want to, before the burrs are mature, you want to cover them up and isolate them from other pollen in the area. So you do this by covering them with a bag and then check on them often. And once the burrs are receptive, you add your intentional male pollen to the bag, give it a shake, maybe add some more pollen a few days later to increase your chances of fertilization. And then you let these sit on the plant. I harvested my crosses in October, so you let them mature in cones for as long as possible. So the next year, so I made these crosses in the summer, harvested the seed in the fall, and then this spring, we will propagate them out in a greenhouse and do disease screening and reduce our population down to about 5,000. So now we're into year 2. So harvest the seeds, plant them out in a greenhouse. This will occur soon here with the USDA material. And then we inoculate with both downy and powdery mildews and cull out individuals that are really, really susceptible. So in the 3rd year, we established these plants on a short trellis for additional disease screening. Also forgot to mention that in year 2, we look at flower morphology and determine sex. And so we'll advance females to the short trellis stage. And then in my case, in year 3, once I have germplasm that's that far along, they'll be screened on a short trellis in Oregon. And this is kind of what that looks like. And then years 4 to 5, the plants will be evaluated in a single hill plot. So John showed some pictures of that, but each individual genotype or each individual plant is a unique genotype as indicated in this little figure by the different color plants. So at this stage, we're around 400 individuals. And you can see in this picture, this is one of John's single hill plot nurseries. You can see lots of variation for the traits that are important for the breeding program. You can see definitely some variation in stature and arm length. So in years 6 through 8, we will take individuals that fared really well in a single hill evaluation, propagate them out into larger trials, ideally replicated trials over multiple environments. We'll evaluate them, maybe initiate some pilot brews. And if something's looking promising at this point, we'll submit it for virus elimination. So here is a picture of an advanced trial in Prosser, as well as some samples that we just sent out for aroma testing and some pilot brews that have taken place in the past. So in years 9 through 11, we're moving to elite trials on farm, and we want to at least trial these for 2 years to make sure that they hold up in a production setting. So do they pick well? Do they train well? How are the yields? And maturity and all of those important traits that John described previously. And then in year 12, maybe 13.7 years, we're looking at releasing the line to the public. So in this figure, I'm showing on the y-axis the proportion of acreage, of hop acreage in the United States that is occupied both by, I'm going to show public and proprietary lines from 1987 onward. And in this case, when I say public, I'm referring to not just USDA release material, but also material that's generally publicly available and not proprietary. So over the last 30 years or so, the acreage of proprietary lines has increased, and the proportion of acreage that's dedicated to public lines has decreased. I think we're all kind of aware of this trend that's going on in acreage. And this project working with Josh Havel from the University of Minnesota. And some of the statistics I'm presenting here are a joint project that we're working on to sort of inventory the public hop breeding program. So historically, the USDA hop breeding program has released primarily cultivars, so lines that are meant to be produced and marketed as hops for brewing. So on the y-axis here, I have the number of varieties. And on the x-axis, I have whether the variety was released as a cultivar for commercial production. There was a time period, I think in like the 70s or the 80s, where there was a major focus on releasing triploid males as pollinators to try to like kind of stimulate yield in the female. So there, Al Honnold released a lot of these lines, and these are male. And then the USDA has also focused a lot on developing germplasm that's publicly available for use in breeding. So a number of these have been female, which is indicated by pink, and then male is indicated by the blue color. So out of a total number of releases is around 63. I believe I have all the records, but sometimes they're a little tricky to come across. And this isn't just USDA alone. This is, in many cases, USDA in collaboration with various experiment stations and universities. But you can see that primarily our releases are based on cultivars, but also that the USDA has done a lot of work in developing germplasm that can support proprietary breeding programs and private breeding programs as well. So the objective is to develop new lines that meet the needs of both growers and brewers and other end users, but also to contribute germplasm that can be used in breeding for everyone. So thanks so much for your attention, and I really look forward to the discussion.
All right, thank you, Kayla and John. Now the next portion of our talk here is we're going to go into kind of a Q&A and general discussion. And I think we can kick it off, I guess, with some questions from the audience. We've got a good crowd here. It looks like we've got about 131 people listening, which tells me the silver lining of an online convention is everyone didn't have as late a night last night and everyone's up bright and early. So the first question in the box here is from Scott Dorsch, and it is Kayla and John, or John, could you please address the concept or existence of transgenic hops and the possibility of commercial applications? Our customers frequently ask about the presence of GMO or genetically engineered hops in their craft beers. Our general response has been transgenic hops do not exist and are not commercially available. Is that still correct, and how much longer will we be able to stand by that statement?
Yes, that's still correct. The only place where any genetic modification of hops is taking place has been in Germany and Japan under experimental conditions. Here in the United States, my program has never done any work with genetic modification in hops. Whether or not this remains that case, I believe pretty strongly that we're not going to be seeing GMO hops in our beer anytime soon. There is some work that's going to be focused upon CRISPR-Cas9, looking at actually modifying disease resistance in hops, but this is only being done to actually test the different resistance genes in hop itself under lab conditions and controlled conditions.
John, for the benefit of the audience, why don't you just real briefly explain what CRISPR is, because it is, you know, the definition of genetic modification can really be a bit broadened, I think, with modern technologies, and it is a much different technology than your traditional GMO or bringing in alien DNA. So why don't you explain just a little bit what you mean by CRISPR-Cas9?
Sure, Jason, that's very true. So CRISPR-Cas9 is something that's just recently been developed, and in fact, the Nobel Prize winner, I can't remember the woman's name, but she developed it about 10 years ago. But essentially what it does is it has it's a system that is designed to attack a certain region of the genome and change that little section of the genome, but not by introducing any new foreign DNA from a different source, but actually creating a little spot mutation. And so it's a very, if you will, sort of a designer system that's very focused. It doesn't introduce alternate DNA from different other genomes, and does hold promise. Now, the bottom line though is the consumer, and if the consumer's not interested in GMOs in their beer, it's definitely not going to take off at this point.
Thanks, John. Another question that's coming up, and I noticed there was one earlier that's related to this, and that is the question of hops in different areas other than the Pacific Northwest. So are there efforts, the two of you foresee efforts of, or increasing your efforts in breeding hops for other regions outside the so-called hop belt of that 45th to 50th parallel, or even other regions within that? I think that there's a couple questions regarding that as well.
Kayla?
Yeah, sure. I can answer that one. I think definitely we want to provide, as a public breeding program, we want to provide hops, like John said, that can be grown all over the country. Obviously, from a plant breeding perspective, that's a very difficult thing to do for every variety. The environmental variation across the country is obviously massive. But I think exposing the plants to, for example, Downy mildew pressure in Oregon and powdery mildew pressure in Washington and so on, we're trying to cover our bases as much as possible to produce lines that can be grown in a wide variety of environments. And then also advanced material, we try to test it in as many places as we can. I know John has some of his advanced lines growing in, for example, Michigan, Minnesota, and you said Vermont too. So obviously we exist here in the Pacific Northwest, but as a public breeding program, we care about developing lines that can be grown more widely.
Yeah.
John, did you have anything to add to that?
Yeah, if I could add to that. So Ann George and I are trying to work together to identify cooperators that we can work with in different states. It doesn't really help us to put these lines out there into different yards, but not have anybody report back to us. And so that's one of our biggest hurdles that we're trying to find. And we're working through that and making progress. The other factor is that any of these lines have to go through the Clean Plant Network before they're distributed. And so that takes a little bit of time too. So we're not going to take a single hill line and then throw it out into the United States. We're going to want to see how it does in an advanced stage before we try and test it. And so once we see it in an advanced stage, then we can get it out to different states. But before that happens, it has to go through the Clean Plant Network.
Right. That's good. One thing I was thinking about as you guys were presenting is the you go through the history of the USDA breeding program, and the releases obviously has had a massive impact on the hop industry at large. Now, with that said, we've had a very big shift, fundamental shift, right, in terms of the customer base and what those breeding targets are. Can you guys comment a bit about that in terms of determining your objectives? You know, John, when you and I first started in this, there was maybe, you could probably count on both hands the number of customers you're really targeting in terms of satisfying their needs, because the makeup of the industry is much different. Now we've got, upwards of, I think it's like 7,000 breweries in the US alone, all of them very creative, doing their own things, looking for different flavors and aromas. So I'd like to hear your perspective on that, John, but then also Kayla, you may have an advantage a bit as with the outsider's perspective coming in to those of us that have been doing this for a long time. So I'd be kind of curious to hear each of your independent viewpoints on that.
Yeah, no, it is true, Jason. I can remember that small handful. It is very challenging, considering that there's 7,000 plus brewers. We are trying to work with Brewers Association to try and reach out to that. And I think the implementation of Brian and Judy as HRC representatives has been phenomenal, because the biggest problem that the public breeding program faces is marketing. And so it's one thing to make a fantastic variety, but if nobody knows about that variety's existence, then it doesn't go anywhere. So that's one of the things that Brian and Judy have been really trying to focus on, is getting the word out. And that's also what Brewers Association has been working on. But it's been challenging. And the market has shifted, because before it was all about developing a line to replace another line. And now it's about developing something that is unique and will be able to allow me to make this style of beer. And so it's a lot more precision sort of breeding and a moving target. Kayla?
Yeah, I think to add to that, I hear a lot about the history of the replacement varieties, like developing an improved version of a previously existing variety, which also in plant breeding is virtually impossible to do, as you both know. And with an outcrossing species, like it's just literally not possible. So I'm kind of glad that the breeding objectives have shifted away from that,
Yes.
just because that's like just a really challenging thing to do. And like Jason said, the room for creativity is really exciting to me, coming in as a new breeder into this species. When I first started, I had a conversation with Ann George, and she suggested identifying varieties that meet the needs of stakeholders at all stages of the supply chain, which I think is a really nice vision for the program. And then there's one trait that I hear a lot about from stakeholders is just something new and something different and something unique. So that's a really cool position to be in as a new breeder, I think.
Yeah. Oh, thank you. I guess John will ask that question. Kayla kind of gave her answer to my next question. But that is, what's your probably the top question you get from brewers or from your stakeholders on the brewing side, or either request or even just general question about breeding in new lines?
So yeah, we still have the, hey, can you make a replacement for Cascade that's higher yielding and disease resistant? But in addition to that, there's requests to build something similar to your Citra, your Mosaics, your Simcoes, these other really popular private varieties. And so that is a big component of the breeding now that we're trying to address. We're not going to be able to repeat what those varieties represent, but we can offer something that's as unique, hopefully, as what those varieties represent.
Mm-hmm. Mm-hmm. Great. I got a couple more questions in the Q&A box here, kind of related to the other side of that stakeholder supply, the supply chain, and it kind of relates back to what we were originally talking about. Well, the first is what are the requirements to become a cooperator in another state or area to report back to the USDA breeding program with feedback? That question comes from Chad.
We'd like to work with a state university, if possible, as a first line. But if that's not possible, then either extension agents would be another person to work with. But in addition to that, we've also worked with private companies or private organizations with one of the brewers possibly as the lead sort of representative to get that information back. But I think the biggest point that we have to make is that it doesn't do us any good to send something out but not get anything back.
Mm-hmm. Along those lines, and this next question relates to that as well. if you consider it from the standpoint to Kayla's point about, having a new release benefit the supply chain from beginning to end all the way to the consumer. This is from Matt Gallagher. Using cashmere as a recent example of a successful public release to the craft brewing industry, what lessons can be learned from its release that can be applied to a USDA breeding direction? Good question.
That's a really good question.
Yeah.
I think what comes up a lot is just whether a variety is noticed and generates excitement. And it's like cashmere has caught on. And I don't know, John, maybe you know more about that variety and can add to that, but.
Yeah, the way that was released is a lesson learned. So essentially at an HRC meeting, Steve Kenny and Eric just said it's released, and that was it. So there was no public notification, there was no generation of interest, there was no announcements, there was no marketing. And so that particular variety just sat around for a couple of years until some growers got a hold on it, and the growers were the ones that actually, and the brewers got excited about it, and it took off from there. But I think the biggest lesson that we've learned from that is that even at the advanced stage and at the elite stage, we need to be promoting these lines. We need to be getting the information out to the brewers, to the marketing companies, just to say, hey, we got this line. You want to try it out? Here's some hop cone to try out and put into your beer. So that's the biggest lesson I would think that we've learned from it.
Okay. Thank you, guys. Let me see here. We got another one from Debbie Thorne, directed directly to Dr. Henning. What is your current thinking for the release of 008 and 074?
If it were left up to me, I would release them right now because, we as USDA scientists, you're viewed on what your publications are and your promotion and so on and so forth. So from a personal standpoint, I'd love to get them out and get that credit. From the realistic side point, I think we need to get a couple more years of yield data, at least one more year of good yield data in both Washington and Oregon, and even possibly Idaho. I think we're going to be trying to get some growing there. But there's a higher expectation, well, not necessarily a higher expectation. All hop varieties that are released today have expectations that they're going to produce well, and we're not going to see any major problems once that variety is released. And so, like I said, I'd love to get something out there right away, but the biggest point is we need to make sure that it's not going to have some unexpected disease problem or yield issues or what have you.
Right. Yeah, that's one of the biggest challenges, right? You go from, as we go through this vetting process, you start with a single plant, move it into a handful of hills, and then go to, even in a commercial planting, it's maybe 1,000 plants or 2,000 plants or hills. That's still, minuscule compared to the eventual expansion. So that's a big challenge. Kayla, I'm going to put you on the spot a bit.
Sure.
So you're new to this, and you and I had a really good chat this summer about your kind of you were trying to, I think, wrap your head around the history, the strategy, where we've come from, and as you've been able to kind of get some experience in our industry, you mentioned all these different strategies for variety development in hops. From the old days of mass selection through pedigree breeding, polyploid, you name it. As based upon what you've seen thus far and your studying of the history, what do you foresee as your preferred strategy? What comment a little bit on your strategy going forward?
Yeah. Well, for those of you who've had a chance to tune into my HRC presentation, I think you will see that I'm a really data-driven person in terms of my breeding philosophy. I like to collect and collect really sound data and make my evaluation decisions based on the data, using like really sound experimental design and lots of environments and things like that. So right now, that's kind of my objective with developing this new breeding program here in Prosser, is really focusing on getting the groundwork in place with regards to some basic things like that. But I'm really interested in genomics assisted breeding, and I really hope to establish that over the next few years. But I really have to like figure out, are the individuals in my field actually who I think they are? You know, and just getting like some of the basic stuff figured out. So, but yeah, I think there's a lot of room for improvement in hops. Just looking at like the crossing record history over the last, like since the breeding programs have been initiated, because of that focus on replacement, there's been a lot of recycling of germplasm. And in plant breeding, we call that like the generation interval. So how long does it take to include a newly developed line as a crossing parent? And this, I haven't calculated the number yet in hops, but something I've been wanting to do, and I suspect is very long. And I think that's kind of like limited our progress in terms of selection to some degree, is because we're just reusing the same stuff all the time. So I'm really interested, and it's something I tried to do in this first year of crossing, is to start including lines that were developed more recently, both as male and female parents. I suspect there's a lot of variation that we can create and select upon and make vast improvements over, you know, the germplasm we've been stuck with since like the 50s. So my philosophy, yeah, and then so I like data. I really also like efficiency, and I think genomics assisted breeding would help with that. For example, I'm interested in predicting the performance of males as parents using genomic data. I think the males are a really underutilized source of genetic variation, or variants that we don't know a lot about the males. And to determine their value based on just phenotype, it takes a very long time. So I'm interested in identifying a genomic prediction approach that can help decrease the generation interval, both in the females, but especially in the males.
No, that's good. I want to come back to the males, because I do think I agree with you. They're kind of an underserved part of the equation. But we have a question that came up, and it actually relates to what you were just talking about in terms of needing to increase the genetic variation within the germplasm pool that's being used for breeding. And I actually had this in my notes to ask a similar question, but this comes from one of the attendees. Wild Manitoban hops, Lupuloides specifically, was introduced into lupulus lineage at some point in the 19th or 20th century. Could you give your best explanation of when and where this occurred, how it has impacted hop breeding in the 20th and 21st century, and phenotypes that can be attributed to this expansion in genomic diversity in commercial hops for beer making? And I think this is a good question, and it relates directly to the diversity question. And it's something I was going to ask too, because there's a lot of talk right now, for example, in Neo-Mexicanus. And the contributions that it's been making of late. But in reality, you know, the use of these wild hops, particularly wild American hops in focused breeding efforts, isn't new. You know, you look back at the history, both Neo-Mexicanus and Lupuloides were used quite early on, particularly in the English breeding program. So if you look at it from the standpoint of Lupuloides and maybe more so recently with Neo-Mexicanus, what do you guys see as the impact of that as a source of genetics and new variation?
I mean, I think it's a real important component, and yeah, you're right, Jason. That was started back in the early 1900s with Dr. Salmon at the Y College there. And that's where we have Brewer's Gold and Bullion and all these other lines that have been used for our super alpha. So basically this wild line from Manitoba, the BB1, that created Brewer's Gold. And all of the super alphas being derived from that. And I know, Jason, you've made a lot of crosses with some of the wild lines, and I know that Steiner has done the same, and I've done the same as well. One of the problems that you run into that is just the marketability of that cross after it's made. Typically, they're very low yielding, and so you have to do some additional breeding steps to be able to utilize that genetic background in your eventual lines that you're going to release. So it's not something that you can easily just say, hey, I'm going to cross with Neomexicanus and then I'm going to get a variety out of it. I mean, there are people that are doing it, but those varieties aren't necessarily going to be high yielding and productive in a real farming situation. But it is something that we as breeders need to be focusing on, not only just for new flavors, but also for pest resistance, for disease resistance, other characteristics, other things that we may not realize that we have to deal with, such as heat tolerance or drought, things like that. So all of the breeding programs are looking into that at this point.
Yeah. Kayla, anything to add with regard to wild sources?
No, that was pretty complete. And I'm personally not super familiar with that quite yet. So
Yeah, no, you are spot on, John, though. That linkage drag in those crosses can be actually pretty incredible.
Yeah.
You know, it can be definitely a 1 step forward, 3 steps back situation when you introduce it into the lines. And so I experienced that definitely personally. We do have another question from the forum here, and it's back to a previous subject we were talking about, but just for the benefit of the participant, are you using or planning to use CRISPR technology? So I guess we talked about what it was, but are you specifically planning on using it in your programs?
So I am not planning on specifically using it, and I know Kayla isn't as well. Oregon State University is starting to do some work with CRISPR with a postdoc that's obtained a grant and will be arriving on campus. We are having them look at some characteristics for disease resistance, but in my lab and in none of the greenhouses and none of the building itself will we be ever having, I should say right now, we will have any material that's CRISPR related. That's all isolated at Oregon State University.
yeah, I have no plans to use it. I think to implement something like that, you have to have a really intricate knowledge of what gene you're trying to alter. And I just don't think, personally, I'm not at that stage in my breeding program.
In terms of technology, Kayla, you've mentioned that one of your goals will be to introduce a genomic approach to breeding. John, you are at the forefront of that in terms of hops and what's going on in the world. Can you guys comment? What are the next steps, or how do you guys see the application of that technology, and what are the latest advances in that work?
Well, for genomic selection, you have to have a really good understanding of the genome and actually have a strong assembly. And so we are about a couple of months away from publishing the 10 chromosomes for the hop genome and complete annotation of all the genes that are present in hop. Kayla's right, we have to have a better idea of what we're actually looking at to make sure that we're looking at the right thing. And so there's some DNA fingerprinting that needs to happen, but I've already started to actually look at some of the quantitative traits and looking at genome-wide association studies for those quantitative traits. I see some strong reason to pursue that. One of the biggest problems that we face is just the DNA extraction step itself is somewhat expensive. And so looking into different resources to try and cut that cost down. But I do see the implementation of genome-wide selection, not necessarily in the greenhouse and seedling stage where we have 60,000 seedlings, but certainly once we get down to the point where we're trying to make decisions as to which of our pots that are in the greenhouse go out into the field, I think at that point is where we could start implementing genome-wide association or just sort of genome selection to help us to pare down that number so that we're not having to look at 5,000 offspring in a field, but maybe just 1,000. But I think the bottom line is that we have to get these things growing out in the field regardless of what sort of method of breeding we do. So we're never going to get rid of our field evaluations at this point. I mean, maybe during Star Trek Future we can just do it, but I think right now we've got quite a ways to go before we're going to be getting rid of any sort of field-based breeding. Kayla, you got anything to add to that?
I would totally agree. Definitely no plans to eliminate field-based breeding to any degree. But yeah, I mean, from my perspective, I'm really excited about just the efficiency of it. In the case of, for example, disease resistance, if we have these known resistance genes, we can genotype a family or a population, for example, and determine which plants actually have the resistance genes, as opposed to like inoculating them and poring over them, trying to evaluate them in the greenhouse over multiple replications and multiple leaves. Like, it's just a lot less work. Obviously, these marker-assisted breeding approaches require validation, definitely. But I think the efficiency aspect of it to me is very exciting. Yeah, but I guess I kind of lost my train of thought. Jason, did you have a specific question for me? I kind of forgot exactly what you were asking.
No, it's just mainly you had mentioned that you want to apply those techniques to your process and your strategy. I just was curious if you had any specific direction you were wanting to take it.
Yeah. So I like, for the example of disease resistance, I think marker-assisted selection would be really useful. But I mentioned previously, I'm really interested in predicting the performance of males. Like, historically, I believe the selection of males has been based on vigor and disease resistance. And then maybe it's tested a few times and like anecdotally or based on some progeny testing, it tends to deliver good female progeny. But I think if we've used so many of these males so many times, we can predict or evaluate the performance of their progeny and try to predict that, associate, those types of phenotypes with genetic variants, and then predict that in the next generation. So I think there's lots of room for gains.
Yeah. Oh, certainly. We mentioned, you mentioned the males, and it's interesting because when I first started in this, Chuck Zimmerman told me that, that old adage that breeding is equal parts art and science. Mm-hmm. For me personally, I quickly learned that male breeding, that is absolutely the case. That's where a lot of that art comes in. Mm-hmm. I think every breeder, we don't have any defined way to look at a male and say, yes, this is a great plant to use. Mm-hmm. John, perhaps you could give a little insight into that as to, over the years that you've been doing this, what do you look for in that? How do you approach the male side of that breeding equation, given they contribute, 50% of the genetic makeup of our progeny?
Yeah, it is a difficulty. And for the audience, just to understand this, male flowers are not like the female flowers. And so we can't look at a male flower and see that it's going to be a super high alpha line. I mean, we have some hints that give that direction, but to say this male is going to produce an incredible aroma line, you can't really predict that at this point. And so what Kayla is saying is, if we could get a better idea of that through genome-wide association studies or other ways to see that would really increase our capabilities in breeding. From my perspective, when I'm looking at a a new male for production, for crossing, I typically like to look at the overall vigor of that male. You know, is it a male that's growing to the top of the trellis and just has long arms, flowers all over the place, and so on and so forth. And as well, it has to have disease resistance. And so these are sort of the things that I'm looking at when I'm looking at males. But then, as already been mentioned, just trying to make sure that we're not making close crosses. And now that I say that, then there are situations that recently where I've been actually making brother-sister crosses. So even that's kind of thrown out the door. But generally speaking, you try to make crosses between males and females that are not genetically similar.
Yeah, thank you. Kind of shifting gears a little bit, something that's been talked quite a bit about, and we alluded to it in terms of the complexity of selecting, particularly for certain traits. You know, you look at something like a complex trait like aroma. It's driven by so many different factors. And in fact, I think as we have come to understand that even though we'll look at the major components of oil, and for historically, we've looked at those as kind of been the drivers of selection in a lot of ways. I think we know now that there are these minor compounds that we are looking at in terms of parts per trillion, rather than a percent, of the actual oils. It's really, they're very impactful at a very small level. And then you throw on top of that there's a strong genetic by environmental interaction with those traits. That drives what a lot of people might call terroir, or just environmental differences. How do you plan as a USDA breeding program to really account for that, or how can you comment on that?
You would ask that question, Jason. Boy, I'm going to add some complexity to that question and just say, what if we select for some of these thiols, these real minor compounds, and we try and increase the levels of that particular compound? What happens to the aroma at that point? Do we make it smell like cat urine and it's not worth pursuing? So I think another component in the future is that we're going to almost have to get to a point where we can, if we're going to be so specific in our breeding objectives, we're going to have to get to a point where we see where is the optimum level of these thiols. Because I know that if you get too high of them, they can become almost repugnant in their flavoring. So, but how do you deal with terroir? I think the biggest thing that I would answer, and Kayla, you can add to this, is just trying to grow things in different regions and just testing them out and making sure we have the chemistry from those regions. How about you, Kayla?
Yeah, I completely agree with the idea of trying to back up a really subjective trait like aroma with something that's measurable. I think that would make it a lot easier to breed for, rather than like, oh, it smelled really good in this field and it smelled citrusy here or floral here. And I mean, as I mentioned, I'm a data type of person. Stuff like that is really hard for me to wrap my head around and quantify. I hear it a lot from different people, and I can't like make a spreadsheet of like aroma. It's really, really, really tricky trait to take a data-driven approach to breed it for. But if it can be explained by thiols or something, that would be really great for me. But yeah, in terms of addressing the G by E or genotype by environment interaction, yeah, testing over multiple environments, having a really good experimental design strategy in terms of replication and commercial checks to kind of be able to account for that seasonal variation. I think those kinds of things would be really helpful. But yeah, aroma is just subjective, and I talk about this a little bit in my HRC presentation, that the lines that you compare certain lines to change every year. And then the rank and performance for aroma changes, and it can depend on when the line is harvested, how it's treated after harvest. It's a very, very tricky trait to breed for. Obviously super important, but definitely is challenging.
Yeah, yeah. I found that my sensory varies by what kind of mood I'm in as well. So
Probably, yeah.
Yeah.
but no, that's good. You know, and it's interesting because in the cannabis world, they talk about entourage effects and, synergies between all these. And that's further complicates it, right? Because you can look at single compounds or a group of compounds and you have to factor in how they're interacting with the rest of them. So it's a very complex issue. I think that definitely, I don't think that's often considered just how complex it really is. So it's job security though, for those of us. So,
Yeah.
we're getting close to the end here. We got a few more minutes. I haven't seen any more questions come up, so I'll answer it with a little bit more generic, or I'll end it with a little bit more of a generic question. That is, looking at it from the entire supply chain, from grower to consumer, or let's, I guess, start think of it in terms of brewers. Wear my grower hat, what can we as growers do to contribute to your success? And on the flip side of that, what can the brewers do to contribute to your success as well, to help us as an industry as we go down this breeding path?
You want to handle that, Kayla, or would you?
Sure. Yeah. Okay. So growers, how can they help? Obviously, willingness to host trials and provide feedback. John and I have been working on establishing a really easy to follow set of protocols in terms of like taking data. And not that we're expecting growers to take a ton of data, but John mentioned like the importance of getting really consistent data back from people who test the varieties is really, really important. Otherwise it's like, you come back with this like evaluation of, yeah, it did really well or it didn't do well, but that's really valuable, but it'd be nice to be able to have some more concrete data. So offering to host trials, providing feedback that's somewhat quantitative in nature would be really helpful. And then from the brewer's perspective, like how can they support the public breeding program? I think willingness to participate in pilot brews, and then again, providing the data back. Judy has been working really hard on organizing that all for us, and her job I think would be easier if it was routine for people just to reply back with a really solid set of results. And I know she's developed, or HRC in collaboration with others, I'm sure, have developed a form to collect that data in a consistent fashion. But then, in a non-COVID era, I welcome anyone who's interested to come help me rub and sniff in the field. I think as a new hop breeder, that would be really helpful for me. Come tell me what you're interested in. And yeah, I think that's kind of been standard here, at least in Prosser, but obviously this year we couldn't do it. So those two things would be helpful to me.
Yeah. Good. John, anything to add to that?
As an old breeder, I agree with Kayla. Just having you come out to the fields and give your input, definitely welcome. I'm always ready to sit down and talk with the grower because we have so many problems that are similar. So yeah, it's good to be able to relate to people.
Mm-hmm.
Yeah. That's a good point. You can't solve a problem if you don't know it exists, and that feedback from stakeholders is absolutely important. So
yeah, can I just add one more thing, Jason?
Absolutely.
Yeah, so you all know, my breeding program is located in Prosser, right in the middle of the Yakima Valley, and there are lots of growers around here. So I also invite not only brewers, but also growers to come check out the germplasm.
Yeah, and speaking from experience, it's a lot of fun. Maybe that's just me being a bit of a geek, but that's my favorite time of year, is to be out in the field, which I'm sure it is for you guys as well. So,
yep.
Well, thank you very much, both of you. This has been a great conversation. I appreciate the presentations and the work that you guys do. I invite the people participating in the convention to view the HRC presentations. And as John and Kayla said, reach out to them anytime if you have any questions. So with that said, thanks again. Next time, coffee's great, but I'd love to sit down when we can over a beer with you guys and have this conversation again.
Yeah.
So
Definitely miss all of you guys.
Yeah. Thanks so much. This was really fun.
Yeah, thanks.
Thank you, guys. All right, so we are going to have a bit of a break. We're going to break from now until about 9:45, I believe, so about 15 minutes. And we invite you to take this time during the break to visit the virtual trade show. A link should be added to the chat. If it hasn't already, it will be here in a bit, I'm sure. So with that said, why don't we take a 15-minute break and we'll see you back here at 9:45.